But do we get one molecule of pyruvic acid from one molecule of glucose? No. The answer is we get two molecules of pyruvic acid. So if we get two molecules of pyruvic acid, then it means that this Krebs cycle will have to take place twice, right? For complete oxidation of one molecule of glucose, the Krebs cycle has to take place twice. And one more thing is going to take place here. See, this GTP will give its phosphate to ADP, right? And ADP will become what? ATP, and this GTP will become GDP again. Hello everyone, my name is Shivansh Gaur and welcome to ATP STEM. I'm here with yet another video on respiration. In my previous video, I talked about glycolysis, and in today's video, I'm going to talk about Krebs cycle. So if you have not watched my video on glycolysis, I would suggest you to go and watch that video first before you go on to watch this video on Krebs cycle, because it will make much more sense to you if you have watched the glycolysis video. Fine. So therefore, I will provide the link of the glycolysis video in the description given below. You can go and watch that video first and then come on to watch this video on Krebs cycle. So here in today's video, we will talk about the Krebs cycle. Now, if you remember, in glycolysis, we saw that from one glucose molecule, two molecules of pyruvic acid are formed, and we also have net gain of 2 ATP. And that process was taking place in the what? Cytoplasm. Right? So see here, pay very close attention. Here in glycolysis, we saw that glucose, which is a 6-carbon compound, it breaks into two molecules of pyruvic acid, and we also get 2 ATP. Where was this process taking place? In the cytoplasm. Okay, now we will see that what happens to this pyruvic acid in aerobic respiration. Because when aerobic respiration takes place, this pyruvic acid later enters into link reaction and Krebs cycle. One more thing, this Krebs cycle was given by a scientist named Hans Krebs, therefore it's called as Krebs cycle. Okay. Now see, the thing is, for Krebs cycle to take place, the pyruvic acid has to be transported from cytoplasm into the mitochondria, because the link reaction, Krebs cycle, and in my later video I will also talk about ATS, that is electron transport system, it all takes place in mitochondria. So it is very obvious that this pyruvic acid has to be transported into what? The mitochondria from the cytoplasm. Okay, so this pyruvic acid enters into the mitochondria. Okay, now before we start with the Krebs cycle, I'm just giving you an overview of the mitochondrial structure. Right now, as you know, that mitochondria is a double membrane bound structure. This is the outer membrane of the mitochondria, and this is the inner membrane of the mitochondria. Okay, and you will see that the inner membrane has a lot of infoldings like this. These infoldings basically increase the surface area, right, which will be helpful in ETS, because the electron transport system will take place in the inner membrane of mitochondria. More the surface area, more the enzymes present, more the complexes present, and more number of ATP can be produced. But we will talk about that in ETS. For now, we will focus on link reaction and Krebs cycle, which takes place in the matrix of mitochondria. Matrix means the ground substance, right? Okay, so the jelly-like matrix which is present here, this is where the link reaction and Krebs cycle will take place. Fine. So it's very obvious that the pyruvic acid which was formed in the cytoplasm, it has to be transported here into the matrix of mitochondria, where the link reaction and Krebs cycle is going to take place. Is it clear? One more thing, these infoldings of the mitochondria, you must have done this before as well, that these infoldings of the mitochondria are called as cristae, or some call it crista, right? So I call it cristae, so it's cristae. Fine. Now, let's see what happens to this pyruvic acid when it enters into the mitochondrial matrix. Okay, so see here, pay very close attention. This pyruvic acid, which is pyruvate here, right? When this pyruvate or pyruvic acid, it enters into the mitochondrial matrix in the presence of coenzyme A, it gets converted into acetyl coenzyme A. This is basically oxidation reaction which is taking place. So pyruvate is getting oxidized to what? Acetyl coenzyme A. In the presence of NAD+, NAD+ here gets reduced. Okay, NAD+ gets reduced to form what? NADH. And very important thing that you can see here, that when pyruvate is getting converted into acetyl coenzyme A, a molecule of carbon dioxide is also released. Therefore, this step is also called as oxidative decarboxylation. Oxidative because pyruvate is getting oxidized, decarboxylation because a carbon from pyruvate is getting removed in the form of carbon dioxide. Okay, so oxidative decarboxylation, that is removal of carbon dioxide. Now it's very obvious that a pyruvate which has 3 carbons, when it undergoes decarboxylation, it loses 1 carbon in the form of carbon dioxide. We get a 2 carbon compound which is acetyl coenzyme A. Right? And this is nothing but it is link reaction. This is called as link reaction. It's very obvious why. See, here we have pyruvic acid which we got from glycolysis. This pyruvic acid has to enter into what? The Krebs cycle here, right? The reaction which links these two reactions, that is glycolysis and Krebs cycle, is this reaction which is taking place. Therefore, it's called as the link reaction. Okay? Once this reaction takes place, the acetyl coenzyme A enters into what? The Krebs cycle. One more very important point that you have to note here is that during this conversion, the enzyme pyruvate dehydrogenase plays a very important role because it removes the hydrogen from the pyruvate and adds this hydrogen to NAD+ to form what? NADH plus H+. Okay, and also magnesium ions are present in this reaction, which also plays a very important role. So do not forget that this link reaction is taking place in the matrix of mitochondria, right? Pyruvate dehydrogenase is an enzyme and magnesium ions are also required for this reaction to take place. After the link reaction, what do we have here? Pay very close attention. It is acetyl coenzyme A, which is a 2-carbon compound. Now this is from here, the Krebs cycle starts. So let's see what happens here. Okay. So acetyl coenzyme A, which is a 2-carbon compound, it reacts with oxaloacetic acid, which is a 4-carbon compound. In the presence of which enzyme? Citrate synthase. Now see here, acetyl coenzyme A, 2-carbon, oxaloacetic acid, 4-carbon. So obviously when these two will combine, they will form a 6-carbon compound. 4 plus 2, 6. Right? So a 6-carbon compound called as citric acid is formed here. Okay, so this citric acid is formed here, which is a 6-carbon compound in the presence of which enzyme? Citrate synthase. Now the next two reactions are going to be very important. Pay very close attention here. Okay, what is going to happen with citric acid? It has 6 carbon compounds, right? Okay, so see, citric acid again undergoes what? Oxidative decarboxylation. Okay, so citric acid gets oxidized to alpha ketogluteric acid, right? And NAD+ gets reduced to form what? NADH plus H+, and 1 carbon from the citric acid is removed in the form of carbon dioxide. Okay, so if 1 carbon is removed, it gives what? A 5-carbon compound called as alpha ketogluteric acid. Okay, please keep noting down where we are getting NADH, where we are getting CO2, right? These steps are going to play very important role later. Okay, so pay very close attention. See, so citric acid, it undergoes oxidative decarboxylation. We have alpha ketogluteric acid, a 5-carbon compound. It again undergoes one more oxidative decarboxylation, and alpha ketogluteric acid removes one CO2. It reduces NAD plus to NADH plus H plus. It itself gets oxidized to succinic acid, and of course one carbon has been removed in the form of CO2. So now we have a 4-carbon compound, succinic acid. But see, something unique is taking place here. See, what is happening here? GDP, and then we get GTP, right? You must be wondering, sir, we have heard what ATP is, what ADP is. Now, what is this GDP, GTP? See, it's just, like adenosine is a sugar, right? Here, guanosine, G stands for guanosine. So it's guanosine diphosphate. Like we have ADP, adenosine diphosphate. We have guanosine diphosphate. Okay, and guanosine diphosphate, it converts into what? GTP. Okay, so here GDP gets converted into GTP, which means a phosphate is getting added to GTP. Diphosphate converts into what? Triphosphate, right? So GTP has 3 phosphates. And one more thing is going to take place here. See, this GTP will give its phosphate to ADP, right? And ADP will become what? ATP, and this GTP will become GDP again. See what happened here. Pay very close attention. GDP here, it got phosphate and it became what? GTP. Then this GTP gave its phosphate to ADP, and then the ADP became what? ATP, and GTP became again GDP. These type of reactions are called as coupled reactions, okay, where one reaction is doing the other reaction. Okay, so this is a coupled reaction, and it's also called a substrate level phosphorylation. Why it's called a substrate level phosphorylation? Because the phosphate to this ADP is not coming as inorganic phosphate, it is coming from any other intermediate or substrate. So when any intermediate, right, when any phosphorylated intermediate give its phosphate to someone else, it's called as substrate level phosphorylation. If inorganic phosphate gets added, it's simple phosphorylation. But here, since an intermediate which has been just phosphorylated here, right, okay, when it gives its phosphate, it's called as substrate level phosphorylation. So ultimately, out of all this, we get ATP, right, and also succinic acid. Can you see this succinic acid, a 4-carbon compound here? Fine. Now, succinic acid, it converts into malic acid, as in it gets oxidized into malic acid, and FAD plus gets reduced to FADH2. Okay, these FADH2 is going to play a very important role in ETS, electron transport system. We will also see that this reaction basically is linked with one of the complexes present in ETS. So again, we will see that later. But for now, what you need to understand is succinic acid gets oxidized to malic acid, and as a result of that, FAD plus is converted into FADH2, right? And it is going to play a very important role in ETS later. Finally, this malic acid, it undergoes one more oxidation, right? And malic acid converts into oxaloacetic acid, right? Oxaloacetic acid is the compound which initiated or which is involved in the first step of Krebs cycle. We need to reform or regenerate this oxaloacetic acid so that this cycle can continue. So therefore, oxaloacetic acid, after undergoing all these reactions, it is regenerated, it is formed again. Why? So that it can enter into the Krebs cycle again. And in this step as well, we get one NADH plus H plus from NADP. plus. So this is the entire Krebs cycle. Now let's quickly see that what are the steps where we get NADH plus H plus. Okay, so at this step we get NADH plus H plus, right? When citric acid converts into alpha ketogluteric acid, here also we get NADH plus H plus. When alpha ketogluteric acid converts into succinic acid, here also we get NADH plus H plus. And when malic acid gets converted into oxaloacetic acid, here also we get NADH plus H plus. Now what is important to note is that this entire cycle that we saw was for one molecule of pyruvic acid, right? But do we get one molecule of pyruvic acid from one molecule of glucose? No, the answer is we get two molecules of pyruvic acid. So if we get two molecules of pyruvic acid, then it means that this Krebs cycle will have to take place twice, right? For complete oxidation of one molecule of glucose, the Krebs cycle has to take place twice. Okay, so therefore we need to multiply everything by 2 if we want to see how much products we are getting from one molecule of glucose. So that is very simple. Let's do that very quickly, right? We need to multiply everything by 2, right? So here we have 2 molecules of pyruvic acid. So here also we will write into 2 molecules of pyruvic acid. So 2 molecules of pyruvic acid will require 2 molecules of coenzyme A, 2 molecules of NAD plus will get converted into 2 molecules of NADH plus H plus, and it will also give out 2 molecules of carbon dioxide to form 2 molecules of acetyl coenzyme A. 2 molecules of acetyl coenzyme A will react with the 2 molecules of oxaloacetic acid to form 2 molecules of citric acid. These 2 molecules of citric acid, they will undergo decarboxylation to give out 2 molecules of carbon dioxide. 2 molecules of NAD plus will get converted into 2 molecules of NADH plus H plus to get 2 molecules of alpha ketogluteric acid. These 2 molecules of alpha ketogluteric acid will also give out 2 molecules of carbon dioxide, and 2 molecules of NAD plus will get converted into 2 molecules of NADH plus H plus. And here 2 molecules of GDP will convert into 2 molecules of GTP. 2 molecules of GTP will convert 2 molecules of ADP to 2 molecules of ATP. Okay, then after this we will get 2 molecules of what? Succinic acid. Okay, so here we will get 2 molecules of succinic acid. 2 molecules of succinic acid will get converted into 2 molecules of malic acid, and 2 molecules of FAD plus will convert into 2 molecules of FADH2. Then 2 molecules of malic acid will convert into 2 molecules of oxaloacetic acid, and 2 molecules of NAD plus will get converted into 2 molecules of NADH plus H plus. So basically I've done nothing, I've just multiplied everything by 2. You don't have to worry about that. Okay, now see what will happen here. Just see total how many NADH we are getting from the link reaction and Krebs cycle. See, it's very simple. Count here. We are getting 2 here, right? So 2. Then we are getting 2 here. So 2 plus 2, 4, right? We are getting 2 here. So 4 plus 2, 6. And we are getting 2 here. So 6 plus 2, 8. Total 8 NADH we got here. So I have written here products of link reaction and Krebs cycle. We get 8 NADH. Also count total how many carbon dioxide molecules we got here. So 2 carbon dioxide molecules we got here in this step. 2 carbon dioxide molecules we got here in this step. 2 molecules in this step as well. 2 plus 2 plus 2, 6. So total 6 molecules of carbon dioxide we also get after link reaction and Krebs cycle. Okay, then how many molecules of ATP we got? Here 2 molecules of ATP we got, right? And how many molecules of FADH2 we got? 2 molecules of FADH2 we got in 2 Krebs cycle. Okay, so that is also something that I have written here. 2 FADH2 we get and 2 ATP we get. So these are the products of link reaction and Krebs cycle. Fine. Now remember, glycolysis have taken place before Krebs cycle and link reaction. So from glycolysis what did we get? We get 2 ATP, 2 NADH. We got 2 pyruvate as well, but that pyruvate got what? utilized in the link reaction and Krebs cycle. So net products of glycolysis here are 2 ATP and 2 NADH. Clear? So till now, from glycolysis and from link reaction and Krebs cycle, the total number of NADH that we have gotten here are 10, which is 2 from glycolysis and 8 from link reaction and Krebs cycle. 2 FADH2 we got from Krebs cycle. 4 ATP we got, 2 from what? Glycolysis and 2 from Krebs cycle. So total 4 ATP and 6 carbon dioxide, which is released into the atmosphere, right? CO2 is released into the atmosphere after respiration. That's what happens, right? Okay. So these are the net products after glycolysis, link reaction and Krebs cycle. Now you must be wondering, sir, that in aerobic respiration, total 38 ATPs are supposed to be produced. That is what we have learned, right? The total 38 ATPs are produced, right? But till now, we have seen the production of only 4 ATP, right? Till now, after talking about glycolysis, link reaction, Krebs cycle, everything we have done, and we have got only what? 4 ATPs. Where are the rest 34 ATPs are going to come from? So these ATP are going to come from the electron transport system, right? See, these NADH and FADH2 that we have gotten here, right? They have a role to play, and they have a very important role to play, right? And their role is in the ETS. Okay, so in the ETS, that is electron transport system video, I will talk about how these 10 NADH and 2 FADH2 that we have gotten till now are going to help in the production of rest 34 ATPs. Subscribe this channel, right, and put on the notifications so that whenever my next video on ETS comes, you get to know how exactly the rest 34 ATPs are going to be produced. Okay, right? But for now, I'm just telling you that the rest 34 ATPs are going to come from electron transport system. Where does electron transport system take place? It takes place in the inner membrane of mitochondria. There are some complexes involved. So we will talk about ETS more in our next video, right? So I hope this Krebs cycle is clear in your mind. The net products of Krebs cycle is clear, the net products of glycolysis are clear, and the total products till now from glycolysis and link reaction and Krebs cycle is clear, right? Let's do 1 or 2 previous year NEET questions to see how much we have learned from this video. Okay, so let's see. The first question that I have, it came in 2018. It says, which of these statements is incorrect? Which of these statements is incorrect? Okay, so let's see. Enzymes of tricarboxylic acid cycle, basically citric acid cycle or Krebs cycle, right, are present in mitochondrial matrix. This is correct statement, right? Okay, we just saw that all this entire process of Krebs cycle takes place in mitochondrial matrix, so this is correct. Glycolysis occurs in cytosol, that is cytoplasm, correct. Glycolysis operates as long as it is supplied with NAD that can pick up hydrogen atoms. This is also correct because NAD was required to take up the hydrogen atoms and we were getting to NADH. So if that NAD is not present, then the glycolysis cannot take place. So this statement is also correct. D statement is oxidative phosphorylation takes place in the outer mitochondrial membrane. No. Oxidative phosphorylation takes place in the matrix of mitochondria. Okay, so here which one is incorrect? That oxidative phosphorylation takes place in outer mitochondrial membrane. So the option, correct answer here is D. Let's look at the next question as well. Which statement is wrong for Krebs cycle? It came in 2017. Such questions comes a lot, right? Based on how many correct statements are there, how many incorrect statements are there. So like this, a lot of questions comes. Okay, let's see. That there are 3 points in the cycle where NAD is reduced to NADH plus H plus. Yes, that's correct. There are 3 places. Yes or no? I was saying now that pay very close attention in which steps the NAD plus is going to NADH is going to produced. See here, 1, 2, 3. So in this cycle there are 3 places. If you consider link reaction as well, then there are 4 places. But link reaction is not the part of the cycle. The cycle starts from here, right? So we will consider only these 3 steps. So the first statement is correct. Okay, there are 3 steps. So let's see the other statement then. There is one point in the cycle where FAD plus is reduced to FADH2. This is also correct, right? Quickly again I am showing you. See here, there is one point in the cycle when the succinic acid gets converted into malic acid, we get FADH2. That is also correct. Okay, fine. Then during conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. This is also correct. Remember here when we were getting succinic acid, right, a molecule of GTP was synthesized. So this is also correct. Okay, so which is the incorrect statement then? Let's read the fourth statement then. The cycle starts with the condensation of acetyl group, that is acetyl coenzyme A, right, with pyruvic acid to yield citric acid. No, right? The condensation of acetyl group, that is acetyl coenzyme A, does not take place with pyruvic acid to yield what? Citric acid. This is incorrect statement. So the correct answer here would again be D, right? D is the correct answer. So if this video was helpful, if after this video Krebs cycle is fully cleared in your mind, right, then please watch my next video, which would be on electron transport system. Right, electron transport system is a little complex process, right? Okay, so do watch that video, because I can guarantee that after watching that video ETS would be super clear in your mind. Okay, so we will meet in the next video. Till then, take care and subscribe. Bye bye.